In the next 7 minutes, we will be solving a question about HIV. I really recommend that you pause the video and try to solve the question by yourself and then try to mark your answer and note your mistakes as I explain the answer. Let's start. Question about HIV. You have 5 minutes to read the question and answer it. 3-mark question. Pause the video now. Alright, so this question is asking about the human immunodeficiency virus, HIV, and it causes AIDS. HIV enters and destroys T helper cells. So they told you what HIV does in the body. Some questions ask, what does HIV do in the body? How does it cause disease? How does it cause AIDS? All right, this is a common question in the exam, and I will not be covering it in this video, but I advise you to go and take a look at this type of questions and try to answer this type of question. The question we're looking at right now, they gave you how HIV causes AIDS by destroying T helper cells. So this is the host cell of HIV. It is the cell that HIV targets and binds to and starts manipulating so that it multiplies inside our bodies. Describe how HIV particles are able to enter. Again, I'm going to stress that you have to read the question very, very well, especially the last statement. The last statement says, describe how it is able to enter. So there will be no point about talking about how HIV multiplies and becomes many virus cells or virus fragments or virus particles, how HIV invades many other cells, how reverse transcriptase converts RNA into DNA. There will be no point talking about that because they asked you in the question specifically to talk about how HIV enters the T cell. So they gave you a limit. Talk about when HIV enters the body till when HIV enters the T cell. After it enters the T cell, I don't want you to talk about anything. So focus. Reading the question is half the job. Alright, so how does HIV enter a T helper cell? You might be thinking, how does it enter? It just binds to it and enters the cell. There's nothing to say. There's no 3 points that I can make up. No, there are. And there are keywords that you need to mention. So let's have a look. I began my answer with a keyword, glycoproteins. This shows the examiner that you know the structure of HIV, and it's very important that you know the structure of HIV. HIV is made up of an envelope, and on that envelope it has a lot of spikes. You can see the picture that they gave you in the question. These spikes are glycoproteins. It uses them to detect cells. All right, so I'm talking about glycoproteins. This is a very important keyword. Glycoproteins on the surface of HIV bind. Again, I'm using the word bind. Whenever I talk about something that comes in contact with a receptor or recognizes a receptor in the topic of immunity, I always use the word bind. So glycoproteins bind to receptors on the surface of the T helper cell. So let's say this is the T helper cell. It has receptors on its surface. These glycoproteins bind to these receptors, and this is how HIV recognizes that this is a T helper cell. This causes the envelope. This is the envelope. All right. Inside the envelope, there is a structure called a capsid. It's not a nucleus. It's a capsid. It's something that just goes around the nucleic material of HIV, which is RNA. So this is the structure of HIV. This causes the envelope of the HIV to fuse. So the envelope, again, you mentioned parts of HIV. Envelope is a technical term that is only in viruses. Viruses do not have a cell membrane or a cell wall. They have an envelope. To fuse with the cell membrane, since this is an animal cell, the T cell is the animal cell, of the T helper cell, and allows the virus to inject. Focus on these words. I used bind, fuse, inject. This is what the virus does. It binds, it fuses, and it injects its RNA. Again, it's important that you recognize or you show the examiner that you know the parts of HIV and the parts of a virus. First, glycoproteins, the spikes, then the envelope, and then the RNA. And you know how to use terms that describe, verbs that describe what is happening. Binds, fuses, injects. This is what's going to get you the mark. Injects its RNA into the T helper cell. Now, it entered. If its RNA entered the T helper cell, the virus did its job. It does not need to enter with its whole envelope. It just injects its nucleic acid or its nucleic material, and that's it. The nucleic material or the RNA does the job. It multiplies inside the T cell. All right, we don't need to mention this because this is as far as the question goes.